years of experience in biologics process development and cell line development, and 12 years of hands-on experience applying next-generation sequencing to advance biopharmaceuticals innovations. She has held scientific leadership roles at several companies. I don't want to read that. Driving progress in cell line development and data-guided process optimization. She is a founder of the Aureli Biologics Solution, a consulting firm focused on enabling next-generation biologics development strategies. Welcome.
Thank you. Yeah, thank you so much for the kind introduction. I'm Christina. I'm currently a consultant for, owning this consulting firm, Aureli Biologics Solution. I'm helping the companies who does cell line development or the needs for protein science to some consulting works. And today I will be talking about the impact of next generation sequencing in the CHO cell line development. NGS technology has been around the field for more than 25 years, but the application of using NGS in the cell line or bioprocess world is a lot less. It's been around 15 years that people have been actively using NGS to define the cell lines or in the other application. But NGS, when the NGS start taking the role, it's been helping us to convert the black box understanding about the protein and move into the high resolution. And we can understand more about the cell line or the protein better because NGS technology, and that can help us to reduce the risk on understanding about this protein because we have this tool. We can detect, early detect the sequence variants when that's even before research cell bank, or master cell bank. So that can reduce a lot of risk. And then there's regulatory alignment. Around 2023, the FDA started being able to accept using NGS to detect virus in master cell bank or on the process box. So that becomes emerging needs for using NGS in the cell line characterization. I'm going to provide an outline of this NGS application. I have 10 things that can use NGS for the CHO cell line characterization. I won't be able to talk every single one due to the time limitation, but I will cover about 90%. First is the sequence variance characterization. So in the protein level, if we are expressing the monoclonal antibody like IgG, we are looking into the heavy chain and light chain and perform a lot of HPLC analysis. And also in deeper level, we can look into peptide mapping. But in the DNA or nucleotide sequence level, we can amplify heavy chain and light chain that got inserted into CHO genome and perform next generation sequencing to look into the sequence variants. The workflow for next generation sequencing and what I've mentioned, that can be a CHO cell lines, and then the genomic DNA from the CHO cell line can be extracted, or RNA that can be extracted and converted into cDNA. Then the amplicom that can be amplified by PCR, amplify specifically heavy chain and light chain based on the size. And there are two different NGS methods that can, or equipment that can be used, like MySeq NGS or PacBio that's perform the long read sequencing and further perform the bioinformatics analysis for sequence alignment. I provide an example from a publication in 2015 They look into the comparison of LC-MS and RNA-seq, or essentially is RNA extracted from the CHO cell line, and then they convert it to cDNA and perform next-gen sequencing. They have found several clones has sequence variants, and this specific clone they highlight in orange that I have the orange box below, showing there is a sequence variance that there is a change from serine to leucine. There's a hydrophilic amino acid residue change to leucine. And in the reverse phase HPLC, they found there's a 2 peaks, and peak 2 essentially is heavy chain peak, and that shows a shoulder, suggests this shoulder is coming from the small sequence variants from the nucleotide level. So they look into the peptide mapping. They also zoom into this specific sequence location and found there is a peptide change in around it's around 1.5% level. So without NGS, it's a lot harder to identify sequence variants using peptide mapping. But with this information, because NGS can run with higher throughput and can go a lot easier and help us to look into a sequence variants easily. So with this tool, it's a lot more efficient to look into the sequence variants. Another technology is TLNA NGS, provided by Sargentis and now been acquired by Solvius. And they are looking into the target locus amplification. Essentially is to amplify the plasmids that got inserted into CHO genome, and also some adjacent gene that belongs to the genomic DNA of CHO, also getting amplified, and they look into, and then they sequence this area, and then got into understand not only the plasmid sequence, but also the genomic sequence around the transgene side. And then further analysis and perform, they can not only know the sequence variants, but also the genomic DNA around it. And this is one of the examples that shows a mutation from methionine to isoleucine, and that's around 33% sequence variance. So I kept talking about the sequence variance in what percent, and like what exactly percentage we are looking into for sequence variance. So I researched about the ICHQ guideline and look into if DNA coding in Q5B, if DNA coding sequence is a stand as the reference genes, then yes, that will be acceptable. And then Q6E, it's more talking about the protein level. The protein molecular variants need to be aligned with the provided sequence. So yeah, it's talking about sequence variants. And Q5E talking about comparability, maybe in the earlier cell line versus the later stage cell line. If there is a change of cell line or even change of the process, then there is a primary structure must be comparable. So we think, all the detection can go maybe as low as 0.05%. But is it really necessary to match that percentage? There's no specific percentage being mentioned in the ICHQ guideline, so we just take it very cautiously. We don't need to reach to that low level of sequence variance. We want, or with super high sensitivity, because some of the bioinformatic analysis, they provide a filter. They might be 5%, or you can change it to a different percentage. But no guideline showing that you need to provide the sequence variance that as low as like maybe 5% or 3%. So we have to take this action cautiously. But yeah, so that helps us become a helping tool, but we cannot overread much. This is a workflow of cell line development from transfection to cell expansion to top 20 clones to the later stage of top 3 to 5 clones, and then perform stability studies. My experience is to perform single sequencing in the earlier stage around maybe top 20 clones, or if there is a concern about sequence variants. But around RCB stage, we can look into using next generation sequencing to detect sequence variants and also compare that information with proteomic data like peptide mapping. Next, I'm going to talk about monoclonality assurance. TLA NGS is a very powerful tool that can determine the integration side, as I just mentioned, that can provide sequence variants information and also the genes that surrounded to your transgenes. And also provide information about breakpoint, and then also how does the structure of the plasmids fall into or goes inserted into the CHO genome. That can be head to tail or tail to tail, or there's a overlap of 2 genes as forms concatenamer, and there's certain deletion between the plasmids. So all this information become a fingerprint for your cell line. Use this fingerprint that can use for molecular methods for monoclonality assurance. If you provide a cell line, the image is not clear, or that's legendary cell line, there's no image from decades ago, then yeah, certainly you can consider looking into monoclonality assurance by these genomic methods. So CHO cells and perform TLA NGS or even whole genome sequencing, and later have bioinformatic analysis. And then use I have a really bottom sequence area, bottom like the gene structure area, and then you can look into the qPCR to amplify this breakpoint region and amplify that by PCR or qPCR and see if you have the parent clone versus the subclone, around 100 clones that you can look into and see if they have the similar pattern like the parent clone. If there are 90% above that shows similar pattern like the parent clone, then they suggest this can be a monoclone. So this will be something that you can consider for regulatory questions if they have concern about your monoclonality. And then this is another application of using NGS to determine the select pool diversity. So TLA NGS can help us determine the amount of integration site. And in this case, when we transfect our gene into the CHO cells, and if there's several different electroporation methods that you use, for example, using MaxCyte or using Neon or using like even the lipofectamine type of methods, then there are various ways of inserting your plasmid genes into the CHO genome. And using TLA NGS, they help you to understand about the amount of integration size around your whole bulk pool. And then if there's a lot more efficient methods, then you got a huge pool diversity that can be maybe thousands, 5,000s, or even more on the integration size. If you have a poor transfection methods, then the amount of integration might be like hundreds. So using this is a interesting methods to help us understand about electroporation methods. And we can use this information to understand if our pool is diverse enough. and we can further modify our electroporation or maybe selection methods. Next, I'm going to talk about epigenetic sequencing and then DNA methylation, and then that's relevant to our CHO cell line stability. The methylation that can happen around the promoter area of our gene of interest, but not necessarily always in the promoter. That can also happen in our gene of interest or poly-A. And the technology of having our sequence, the methylation part of the genes is not on the nucleotide itself anymore. It's more like on the top of that. So there is a cytosine that if that is getting methylated, and then we have a toolbox that can called bisulfite conversion, and that can convert cytosine or non-methylated cytosine to uridine. And we can perform PCR amplification and look at all the sequence, compare with the reference genes, and understand about the percentage of methylation. So I provide one of the literature from 2015 and 2010 talking about the methylation on the CPG islands of genes around this monoclonal antibody. So when there is a methylation level below 20%, then let's consider it's a light methylation or light silencing. When there is 20 to 60%, then this methylation is more like medium level. But if the methylation is larger than 60%, then that is called hypermethylation. Another example in here is using this antibody productivity to look into if in the early stage it is black. Is a black bar, and the later stage is a gray bar, showing like the clone number 2 is in the second set of bars, and then clone number 3, that's in the third bars, showing they are relatively having less stability compared to the clone number 1 and then clone number 4. Or here's clone 7 and clone 11. So 9 and 2 is slightly comparatively less stable, and they look into the methylation with and without selection, and they do find without selection, then they do find the methylation level is higher in the clone number 2 and 3. And if they added the methylation inhibitor, it's called 5-ZA-DC, then they found, or 5-ZA-DC, the methyltransferase inhibitor, then they found the methylation can be reduced. So this suggests methylation can be detected by this epigenetic sequencing methods, and that can be impacted by, that can impact the stability of the cell clones. Next, I'm going to talk about the viral safety advantageous agent detection using NGS. As FDA recently, around 2013, October, they accept using NGS as a method to detect virus in the master cell bank or end-of-process cell banks or even the unprocessed bulk. So that in the viral testing field, it used to be using in vivo or animal tests to detect virus, but that becomes a lot longer process, and then animal use can be less desirable in these days. And so there are also in vitro tests using cell assays to detect if the virus is present, and this become more like standard till now. But these 2 years, NGS started to become another alternative for the viral testing. And that can be animal-free and then more precise in sequence identification, and that's become an emerging technology to replace in vivo, but it's an alternative for in vitro assay. But using NGS, we need to be very cautious because next generation sequencing is determined in the sequence in the DNA or nucleotide level, and it's not necessarily telling you which one is a live virus and which one is endogenous virus. Endogenous virus is the gene that already present in your host cells for a long time, and that's not an active virus. So we need to be very careful about this, and we can also be very careful about using the vendor who provide the viral testing. They have to reduce the background interference, such as environment contamination, that can cause some of your tests not accurate. And and then, of course, look into the vendor who perform viral testing. We need to make sure they are GMP scale, and they have a standardized bioinformatics tools that can interpret your process, your virus well. Like, for example, they need to have a great bioinformatic tool. They use the proper or not out-of-date database for your viral testing or virus sequence alignment, and they need to have a sequence alignment tool that can do all the analysis properly. So we want to be very careful about non-infectious and irrelevant viral sequence, and that's part of the risk. So in the regulatory position and strategy risk, we want to generate data that's reliable, not that shouldn't be able to that should be able to explain properly. Sometimes we generate data that's not cannot be interpreted properly, then it can trigger a lot of investigation, and we don't want that. So I think the partnering between NGS and an in vivo testing might be a good idea. So we want to use this cautiously. That's more like the next generation sequencing, emerging technology for viral detection. Finally, I would like to talk about transcriptomic sequencing and use this as a tool for whole cell protein characterization. I won't be able to talk about CRISPR for application. But I would cover what we really think that's a very important part of the NGS application in the cell line. In whole cell protein, I think we have been widely accepted by using the LC-MS method to understand about the abundance of whole cell protein in our protein products. And that will be part of the later stage of clinical documentation need to be provided in the later stage. And then in here, this is a publication 2024 and 2025. They use both methods they used LC-MS to help determine the whole cell proteins and then showing their abundance in the 14 days of HCCF fat batch culture. And another interesting application of using RNA-seq, and they use all various kinds of cell clones that express different type of antibody and look into the processes like different culture days, and they start showing difference of sequence. It shows different abundance of whole cell protein at different culturing day. And this tool becomes a lot easier for us to understand about whole cell protein expression at different days and different clones. So it's the NGS become a lot faster, high throughput for us to understand about the cell line or cell culture performance. So I think the combination of proteomic and transcriptomic methods that help us to profile the CHO cell whole cell protein, and then using proteomic methods, a profound tool that we can investigate the manufacturing process such as fed batch or perfusion. And then using transcriptomic profiling that help us understand cell viability is not the only predictor anymore for whole cell protein, but that we can look into the clone, the cell line, media, process parameters, and more. All these things can become very helpful. And then, I mean, using the transcriptomic methods to help us look into HCP more clearly. So I would like to have the summary of this talk is the impact of NGS of cell line development. We utilize NGS and provide a more proactive quality control that can early detect the sequence variants from our cell line, and that being be a orthogonal control tool to help combine with NGS and peptide mapping for sequence verification. And then monoclonality assurance is a very important part of the regulatory documentation to provide. So this is a very important tool for us when our monoclone image is not clear. And viral safety advantageous agent testing is also a very important, and NGS become a very important tool, or that can partner with in vivo testing to provide the evidence of virus safety. Multi-omic integration that combine proteomic, genomic, epigenetic sequencing, all combined that can help us understand better about our cell culture behavior. And then early stage of screening. I mean, this is a great tool for early stage screening and predict the sequence variants. Yeah, so this concludes my talk, and then thank you very much for inviting me for this presentation. And I would love to have questions from the audience. Thank you.
Thank you for the nice talk. But we only have time for one question.
Maybe I have a silly question. So, with respect to DNA methylation, right? DNA methylation induced silencing of transgenes. And if you prove that it is because of DNA methylation, can it be stated as a stable cell line in later regulatory stages?
Oh, you're talking about the methylation that caused the silencing of the genes?
Yes.
And then that can be later in life?
It is reversible, right? If you treat it to 5-ASA, it is reversible.
Yeah, sometimes it's not reversible. Like if you are continuing the same process and then perform that in for example, the media you use for stability assessments, then that probably cannot be reversible. Yeah.
So it's considered unstable.
Right. Yeah. But if you added, like in the paper I mentioned, they used the methylation inhibitor, then they started from very early. Then that's a case that shows less methylation found.
Thank you.
Thank you. Thank you very much.
Thank you.